Okay, let's talk about the different types of vaccines that we have. So some of the most common ones are inactivated or killed vaccines. And basically what this means is that the microorganism is killed. Along this line of inactivated or killed vaccines, we also have bacterian, and this would be bacteria that has been inactivated. This only produces a short-term amount of immunity, only about a year. And then we have toxoids, and basically toxoids, they stimulate the immune system. They have a lot of adjuvants, which we're going to talk about in a little bit. Okay, this would be your tetanus vaccine. Okay. So what are the pros and cons to using inactivated or killed viruses? Well, let's talk about the pros first. The pros are there's little risk of virulence. So the chances of the disease becoming active once you give the vaccine are pretty small. There's no risk of shedding. So say the animal defecates and the vaccine is not inactive, so it's not going to spread to unvaccinated animals. It's more stable. You still need to handle it with care, but you can be a little bit more, it's a little more forgiving. Generally, you don't have to mix it. It comes already pre-packaged in a vial. It could be multiple dose vials. And if you do have to give a vaccine to an animal that's pregnant, there is a less chance of causing abortion in that animal. Now the cons. The cons of using a killed vaccine are that it needs at least 2 initial doses, and these doses need to be 10 days, at least 10 days apart. With the rabies vaccine, we give a dose at 16 weeks, then we give a booster dose at a year, and then depending on the state you're in, you may not need to vaccinate again for another 3 years. But again, there's the 2 initial doses there. It is a slower immune system response. It takes about 2 to 3 weeks for that immune system to kick in, and it might not remember it. Okay? It's generally tends to be more expensive to manufacture. And because of this problem of the slower immune system response and a weaker immune system response, we need to add adjuvants. Adjuvants help to increase the response of the immune system. It helps enhance it. But the problem is that adjuvants cause cancer. They may cause cancer. They can cause more hypersensitivity. This is one of the reasons why when we give rabies vaccines, we always give it in the rear leg, the right rear leg, because we want to know where we've given that vaccine that is a killed vaccine because of the chance of getting a fibrosarcoma. Another type of vaccine that you're going to see in clinic, modified live or attenuated. And basically what a modified live is they have either genetically manipulated or chemically manipulated or used cultures to manipulate the virulence of this disease. So they lose some of their virulence. It's not necessarily avirulent, but it loses most of its virulence. The pros of this are generally 1 dose is sufficient to provide protection immediately. Okay? It's less reactions, doesn't have any adjuvants in it. It has prolonged protection. This is because it stimulates the T cells and the B cells quicker, which falls in line with this better immune response, and they tend to be more economical. The cons are, well, we're losing virulence, but it can become virulent. Okay? It can break down. It can become virulent. It can cause the disease. Hopefully the disease is more controllable, but it can happen. There could be an excessive immune response, okay? So they could still have some sort of an allergic reaction. Their immune system could just over-respond to it. It is less stable than a killed vaccine. You need to be more careful when you are reconstituting and storing it. And there is a greater chance of abortion risk. There's a little star here. Just because modified live vaccines provide quick protection, they do need to have boosters in order to keep that protection going, to make sure the memory lasts. Live vaccines are very similar to modified live in pros and cons. The thing with live vaccines is, 1, they provide long-lasting immunity, but there is a risk of the disease spreading. and not only to the other animals, but to the humans who administer them. So, recombinant. Basically, a recombinant vaccine is a microorganism that has been recombined. They've done some genetic manipulation. So there's 3 categories of recombinant vaccines. The first one is genetic cloning. So basically what they do is they take the virus DNA, they take parts of it, they put it in a donor cell, they then extract the purified antigen, and they turn that into a vaccine. We're talking about genetic cloning here, so these vaccines tend to be expensive. Category 2, I should have a 2 there, is genetically attenuated. And basically what they do is they take bacteria or bacterium, they take the DNA and they delete the genes that cause the virulence, okay? Then they take that, turn it into a vaccine. The third type is live recombinant, and basically what they do is they select the parts of the virus that stimulates the immune response, and they turn that into the vaccine. There's only one one vaccine that's been approved in the United States by the FDA, and that would be the Newcastle vaccine, the Newcastle virus vaccine. That is your ticket into class. And the final type of vaccine, the final few types of vaccines that we're going to talk about very quickly, polypeptide vaccines. Basically what they do is they have the bacteria or the virus, they insert foreign DNA. Okay, this is why I don't grade on spelling. They have a vector, okay? So this turns it into a vector. They give it IM to an animal. They draw blood. They get the plasma from there, or the proteins, the antibodies, and they turn that into a vaccine. Then we have anti-toxins. Anti-toxins are antibodies that they get from a hypersensitized animal. So basically they give the disease to the animal, they pull blood, and they pull the proteins from that and turn it into a vaccine. This is passive immunity, so it is short term. Then we have anti-serum, and basically it can come from either a hypersensitive animal or it can come from an animal that has had the disease. They again pull blood, they pull off the plasma, and they turn that into a vaccine. If you were to get tetanus, after you are worried about after the tetanus, can give you an anti-serum vaccine, and that is basically what this is. It's from a sensitized animal. And then there's autogenous, and this is a vaccine that's developed from an ill animal. This tends to happen in herds where all of a sudden a disease pops up. They'll take the disease from a sick animal, then they'll send it to a manufacturer or who then changes it into a vaccine, and then they'll vaccinate the whole herd. Don't forget, Newcastle disease is your ticket into class.